Hello, everybody, and welcome to the second part of the Alzheimer's presentation. My name is Sarah Perry, and in this presentation, I am going to discuss the biomarkers for Alzheimer's, which are molecules that are frequent in Alzheimer's, as well as the biochemistry for these biomarkers, the theoretical pathways for Alzheimer's, and the current and experimental treatments for Alzheimer's. But before we dive into all of that, I'm going to quickly give a few reminders from my last presentation on the basics of Alzheimer's. Now, Alzheimer's is the most common form of dementia, and as a quick reminder, dementia is an umbrella term for any memory problem. And I'm not simply talking about forgetting a question on a test. I am talking serious memory problems in which someone might leave an item in an unusual place, forget a name or place they normally would not be able to forget, etc. It is a progressive neurodegenerative disease, which means it targets the neurons in the brain and destroys them over a period of time, which means it does not get better, it only gets worse over time. It occurs mostly in the elderly, which is defined as people who are 65 or older, especially in women, though this is believed to be due to the fact that women statistically outlive men. It is not contagious, but based off of genetics and environmental factors such as obesity and any type of brain damage the patient might have suffered. Diagnosis is based off of memory testing and interviews from friends and family regarding any behavioral changes, though CAT scans and MRIs may also reveal any changes in brain density, which would also lead to a diagnosis. However, as of right now, the only way to have a conclusive diagnosis of Alzheimer's is through an autopsy of the brain. Now on to what you've all been waiting for. There are two main biomarkers for Alzheimer's: beta amyloid plaques and tau tangles, also known as neurofibrillary tangles. Now, in the picture to the right, you can see a comparison of a normal neuron to that of a beta amyloid plaque and a tau tangle. And you can see how the neuron with the beta amyloid plaque and the tau tangles, how it is swollen in the axon, as well as how the synapses are blocked. In a few moments, we're going to go a little deeper into that. Now, amyloid beta fibrils are what most scientists focus on when researching Alzheimer's. Amyloid beta peptides are cleaved from the precursor APP, which is a internal membrane protein. aggregation of amyloid beta fibrils, which is another way of saying the accumulation of amyloid beta fibrils in the brain, causes plaques. These plaques form in the synapse of neurons. So, going back to the picture, you can see how these plaques are blocking the synapse. And as a result of this blockage, communication between the neurons is blocked and the neurons die. Hence why, and because the neurons die, the brain loses that mass. Something else that is interesting is that studies have indicated that when the amyloid beta plaques bind with iron, it forms a neurotoxicity, which is basically a release of reactive oxygen species in some cases. And the reactive oxygen species causes neurological damage. However, this is only one possible way that amyloid beta plaques can cause Alzheimer's. In a few minutes, we're going to be going over other possible pathways. And something else to keep in mind is that our bodies naturally have iron due to red blood cells. Remember, that is why red blood cells are red, is because of the iron within them. So, preventing the binding of amyloid beta plaques and iron would be difficult, but not impossible. Now, as stated earlier, the aggregation of the amyloid beta fibrils in the brain is what causes the plaques. Now, there are two types of amyloid beta fibrils that scientists are looking at. Amyloid beta 40 and amyloid beta 42, though most focus on the 42. Aggregation of these two causes apoptosis of the neurons, inflammation in the neurons, because recall how the neuron right here is swollen. Though we'll go into the other reason why it's swollen in a few minutes. Synaptic degeneration, memory deficiency, all of which collectively lead to the regeneration response, including stem cell activation and neurogenesis. Now on to the tau tangles. Normally, it is a proton within the axon of the neuron that stabilizes microtubules. It is produced through alternative splicing of a single gene called MAPT. The phosphorylation of tau leads it to clumping to other tau proteins, causing the tau tangles. Though it is important to note that it's not just the phosphorylation of tau that leads to clumping. It's also the hyperproduction of tau from the MAPT gene. And interesting enough, this is caused by the amyloid beta plaque. Now, something I forgot to mention earlier is that amyloid plaques, even after they form in the synapse, it can take decades for symptoms of Alzheimer's to appear. In fact, there was one study that showed it can take as long as 22 years for symptoms to occur after the plaques form. And something else that is interesting is that the tau tangles form years after the amyloid beta plaques form. Though this is this is believed to be due to the fact that the amyloid beta plaques do cause, or possibly cause, I should say, the clumping of the tau proteins into the tau tangles. Though it is interesting that the tau tangles form years after the amyloid beta plaques. Though when the tau tangles form is usually a good indication of that the symptoms of Alzheimer's will soon appear, though there is no way to know that until the symptoms of Alzheimer's appears, as of right now. Now, something that scientists are still trying to figure out is if neuroinflammation leads to tau phosphorylation, or if phosphorylated tau leads to neuron inflammation. Now, I know that I said just a few moments ago of how the amyloid beta leads to the tau phosphorylation, which is true, though recall that there are several theoretical pathways in which a lot of these pathways agree that the formation of the amyloid beta plaques is a precursor to the development of the tau tangles, though it's unknown as of this moment if the direct precursor to the tau tangles is neuron inflammation or tau phosphorylation. One of the reasons that scientists hope to figure out the exact order is because scientists will prefer to develop a treatment for the earliest step. Thus, one of the reasons why they are focusing on the amyloid beta and trying to clear that. Though I'll be going more into that in a few minutes. Though I do find that quite interesting of how they are not sure. In some ways, this is kind of like the chicken or the egg question, if you think about it. And here are some of the theoretical pathways for Alzheimer's. So you can see of how the amyloid beta is usually a precursor for many of the pathways, including for the tau tangles and the reactive oxygen species and inflammation. Though keep in mind that, as I said many times before, these are simply theoretical, based off of current evidence. Because in fact, earlier this year, scientists have found another possible pathway for Alzheimer's disease in mice as they were, I believe as they were looking at possible cancer treatments, which they are, and they're currently calling this new pathway the LADN pathway, I believe. So, and though in regards to these pathways, one of the main receptors that scientists are looking at right now is actually the NMDAR receptor. So the reason I say this is because most of the research papers that I've come across discuss it as one of the causes of, well, not quite the cause of Alzheimer's, but rather it is one of the some of the experimental treatments are focusing on inhibiting it. But I'll go more into that later. Now, as for treatments. As I said in my first presentation, there is currently no cure for Alzheimer's.